Let's now consider myoglobin as an example of protein structure and see how it relates to the function of this important protein. Myoglobin has only one polypeptide chain and therefore does not exhibit quaternary structure. It contains 153 amino acids and one heme prosthetic group. To understand the significance of a prosthetic group, consider prosthetic limb. It is something that is added after the protein is made, but is essential for function. We'll look at the structure of heme in a moment. For now, I'll just mention that it is an iron-containing compound essential to its role in oxygen binding. From the illustration here, hopefully you can see that the heme group is buried in a protective hydrophobic pocket. Myoglobin is very compact and is entirely alpha helical in secondary structure, with 8 separate alpha helices. As is true for most globular proteins, the polar side chains of myoglobin are on the exterior surface and the nonpolar residues are on the interior. There are 2 very key histidine residues that interact with the heme group and influence its ability to bind oxygen. Examining the structure of the heme group, we find that it contains 4 5-membered rings based on the structure of pyrrole, and these are linked by methane groups. Its chemical structure is referred to as protoporphyrin 9. Let's look more closely at how the heme group is coordinated within the myoglobin molecule and how this relates to its ability to bind oxygen. At the center of the group is an iron 2 atom, and it is this iron atom that directly binds oxygen. Keeping this iron in a +2 oxidation state is essential for binding to oxygen. This relates to why the heme group is buried within a protective hydrophobic pocket. Notice in the figure that the central iron atom is coordinated to 4 nitrogen atoms within the 4 pyrrole rings. These are pictured as blue spheres. A 5th coordination is to one of the nitrogen atoms of a histidine residue below the plane of the heme ring. This is noted as histidine F8 because it occupies the 8th position within the 6th or F alpha helix. These 5 connections to iron are always present. There is a 6th coordination site to histidine above the plane of the heme ring at position number 7 within the 5th or E helix. Note that this 6th coordination exists between histidine E7 and the bound oxygen. It does not directly connect to the heme iron and exists only when myoglobin is bound to oxygen. We find that more than one type of oxygen-containing molecule can bind to the heme iron. The affinity with which the heme group binds carbon monoxide is 25,000 times that of oxygen. Look at the figure on the far right, and you can see that the position of histidine E7 forces the oxygen molecule into an angled binding to the heme iron. In the center figure, we see that the same is true when heme binds carbon monoxide. To see the significance of this, look at the figure on the left. If the histidine E7 residue were not in position, a carbon monoxide molecule could bind to the heme iron in a linear alignment, enhancing the binding and its deleterious effects. Note that carbon monoxide is toxic and is produced in trace amounts during metabolism, so this type of protection is essential. The example of myoglobin reviewed here helps us see the very close connection between native conformation and protein function.